Welcome to the part 2 of the lecture. So in this part, I'm going to introduce you to Mega software. Using the Mega software, you can analyze your own data which you generated in the previous part, so playing the game, mutational board game. To explain some basic principles of phylogenetics, and in particular use of the program Mega, I created a test data set. So here you can recognize our original pulse, our original sequence. And in the sequence 1, so I designated as sequence 0. In sequence 1, I have replaced one nucleotide, A to G. So here we have one substitution out of 10. Here there are 10. In the sequence 2, I substituted 2 nucleotides. These 2 nucleotides are now different. In the sequence 3, I substituted 3. In the sequence 4, so I would like to put G in this position as well to make it 4. However, here is the G, so I changed it to T. And in the final end of sequence 5, I substituted it to G again. So here there are 5 substitutions in comparison to sequence S0. So now let us analyze these sequences. So here I have on the data tab, pull down menu, I have phylogenetic analysis. So it asks whether it's protein or nucleotide sequence, coding sequence. It's not protein coding sequence. Okay, so here I have the data set appears. So if we click on this tab, we've got a sequence data explorer, and we see the difference between our 5 test sequences. So here they are identical in this area, and here they are different. They differ, and here there's a consensus sequence, so to say. All right, good. Let me start with the distance. So genetic distance between them, and we compute pairwise distances. So I click here. Now I can look at this options what we have here. So scope is our pair of taxes. So we are not going to use sophisticated bootstrapping, so stability tests and reliability tests for now. But let's say, so we will put it here, none. Substitution type, nucleotides. So we're going to play with nucleotides. And the models. In the models, we choose p distance. What is the p distance? So there are quite a number of choices here, quite sophisticated. And then p distance is the simplest choice. Let's check what is that. So p distance. Here it is. So p distance is equal to the number of mutations in the sequence in comparison to the other sequence, the reference sequence, divided by the length of this sequence. So as simple as that. For example, so if we have one nucleotide substituted in one sequence in comparison to the another one sequence. So we got 1, and since the length is 10, so the result will be then 0.1 if there's a 1 substitution. So if there are 2 substitutions, we've got 0.2 as the p distance in here. All right, so substitution types. There's a variety here of the substitution types. We are going to count everything, which is transitions and transversions. So pyrimidine to purine or pyrimidine to pyrimidine. So these are transitions or transversions. So we are going to use uniform rates, and this pairwise deletion we don't have gaps. So, but we come to this point later. So let us calculate this distance. So what do we have in this distance matrix? So we have the pairwise distances between all the 5 sequences, and as we remember, so there's a between first and the second sequence, there's only one nucleotide substitution. So therefore, the EP distance is 0.1, 1 divided by 10. So if we multiply this by 10, it will give us the number of the substitutions, so 1. And we see that the numbers are pretty okay, so reliably reproducing our changes in the sequences. So 1, 2, 3, 4, and then so on. And then within the rest, between let's say 3 and 4, we've got quite consistent numbers as well. Okay, so these numbers represent the number of substitutions or genetic distance between sequences. All right, so let's build the evolutionary relationships between these test sequences in the form of phylogeny. So here I can select phylogeny, and tap here phylogeny. So I click here, and then in Mega we have a choice of 5 methods used to reconstruct these phylogenies. So they were developed in the different time periods for different applications, and some of the feature-based, so some of the substitution-based, and we are going to have a short video explaining the how one or another method works later. Let's use the first one, which is maximum likelihood tree. So click here, and we've got again the palette of the options. So the method which we're going to use is going to be maximum likelihood method. And now we're going to test our phylogeny. So we're going to run it as the bootstrap methods. So what does it mean, bootstrap? We're going to run this method 500 times, not one time, but 500 times. So we're going to get 500 trees. So these trees are then analyzed together, and a consensus tree is produced together with the number. And this number represents consistency or reliability of particular branch in the tree. You will see it in a minute, and I will explain it there. So all the rest, all the other choices are pretty obvious. So how uniform rates of the mutations. Then we use all sites in the analysis. So we click. So we are going to get, so the counting is going here. So we are going to reach 500. So these are going to be our number of bootstrapping. Okay, almost there. All right, this is our tree. So let's look at the right side first. So the good news is that we have correct order of our sequences. S1, S2, S3, S4, S5. However, this branching pattern doesn't make any sense for us. And the reason is that we have to put the original or root of the tree somewhere here, because this tree is not rooted. So maximum likelihood method doesn't give you a meaningful root. Okay, let's place a root. So to do this, we have to select the branch, because we know that we span all our sequences from the S0. So I selected this branch, and here there's a button just called root the tree. Now I rooted the tree. Now it makes sense. Okay, here we are. So this is our seeding sequence. From it, all the other sequences in here were produced by substitutions. So all these sequences are produced by substitutions. Yeah. So let's select them. like this. Okay. And the first sequence which we produced was S1. And from this sequence S1, all the other sequences were produced. Like this. Then we look inside blue rectangle, and we note that from this sequence S2, the rest, S3, S5, S4, S4 were produced. So they were produced from here. And then we look at this sequence S3 inside. And very obviously, so it was produced, all the rest was produced. So from here. So therefore, it makes sense. This branching pattern, which we observed in here. So now bootstrapping. So the reliability of the tree reconstruction can be inferred from the bootstrapping number. Say, for example, this one, 72. 72 means percent. Out of 500 reconstructions, 72% converged on this particular branching point. So they agreed on this branching. So then the next branching is also supported statistically. 70%. So usually like something above 50% in cladistics consider it as reliable or kind of acceptable bootstrapping. And here there's a 71. So more or less, here I don't see the number. Most likely it's technical something. So, but these numbers are telling us that this reconstruction is reliable statistically. So, and it perfectly makes sense. The branch lengths, so they represent the number of variation in here. So, but in the reversed order. So we got so many variations in this sequence. So let me try another methods like fast methods. These three are fast methods, and these two are relatively slower. So here again. I have an option, so options, I will do bootstrapping. Yeah, it's practically the same, but neighbor joining. So we're going to get some information about this method later. So, and again, you can see it's fast. So we quickly get reconstructions. And we get, again, correct order of the sequences. And again, here, I will route it. So now we got higher, so in the neighbor joining method, we got higher percentage of bootstrapping. So you can see, but otherwise the tree is primarily the same. So discard result, and then in the minimum evolution. So the setup is the same, just the method is different. So let me calculate it again. Okay, so minimum evolution methods results in this similar representation, similar tree. So I can actually reorder it. Yeah, and then we get high numbers of bootstrapping again. So let me try UPGMA method. And all right, so here in this particular method, I can't route it because it's constructed intrinsically rooted. Root is calculated as the point in the tree from which all the distances are the same to the all autotaxonomic units. And here, yeah, this is not particularly the same. So because this should be, the root should be right here. And also we have the order of the grouping is not correct because it should be like 0, 1, 3, and so on. Yeah. So, but this is easy to fix. So I can just rotate the branches. You see subtrees like this. I select a branch and then I rotate it. So now if I click here and then I rotate these two groups around, okay, and now here Okay, 0, 1, 2, 3, and here 4 and 5, so I just rotate it here. That's it. So the order of the grouping is the same, and then if we can root this tree, so we basically can place the root here, and then it will look exactly like the previous trees. So, and then finally, let's look at the maximum parsimony. So in the maximum parsimony method, so yeah, it says that in 13 out of 500 replicas we can't reach solution. So, but this is not a big deal in here. So again, we just simply put the root in the right place, and then we've got all the tree and correct here. So 0, 1, 2, 3, 4, 5 with the high bootstrapping numbers. So our method of choice to reconstruct the trees would be then one of the fastest methods, so with the highest bootstrapping. So from this group, Okay, let me revert back to my original game. So it's here I get F3. So I generated 8 sequences starting from this original one. So now the question would be then, so what is the genetic distance? The genetic distance is the number of substitutions, as we discussed, right? So here at every step we have one substitution exactly introduced. So how we can interpret this genetic distance? Genetic distance would be then the number of steps down the tree and up the tree. Up. So from here, from this sequence to this sequence, there are 1, 2, 3, and 4 steps. So, and therefore, these are 4 generations, and therefore it's 2 generations away, because we travel it back and forth along different passes. So we have to divide it, the number of steps by 2. 4 steps divided by 2 generations. So this separated from this sequence by 2 generations, from this sequence sorted by 2 generations. Yeah. So, and what about this? Is the same. So it's 1, 2, 3, 4, 5, 6. 6 divided by 2, 3, by 3 generations. Therefore, it's called F3. Right? So, therefore, looking at the number of the variation within the pulse sequence pairs, we can tell how many generations ago they had a common ancestor. So that's how we can interpret the genetic distance in here. So let me pull up Mega again with the sequence which we have just considered. And then I would like to comment on these missing gaps in here in the sequence, which is quite important aspect here. Let me first store it. So capture it, this one, sequence, and put it aside here. So now, as I said, we have kept the gaps inside as the phylogenetic signal. However, when we do sequencing, for example, like using Illumina or any other methods, so we can't just simply like read the gap in the sequence, because there's no gap in the sequence. So in the real one, what we have is this. So we have to select, and here there's an option which is called delete gaps. So that is what we usually get out of the sequencing machine. And you can see some positions are aligned now, still like A's on the top of C's. However, here we've got the shift to the left and we got misalignment. So now we're going to compare C with the T, which Actually, T should appear in this position, number 3. So this is alignment problem. And this is one of the central problems here in the phylogenetics, how to properly align the sequences. And the more divergent sequences, more difficult to align them. So here, this alignment. We can choose realign those sequences back. Okay, so let's realign them. We click here, then we align DNA. And then it says nothing selected. Select all. Yes, let's select all. Selected all, and this is the method of alignment. Again, there are some parameters, but just let's use this as they are. So, okay, now we've got gaps reintroduced in here. Oh, the question is whether this reintroduction is kind of like correct. Let's see. Okay, this is our original alignment. And you can see here we've got comparable alignment. So let me draw it here. Yeah, quite okay. However, here the gap comes at the end of the sequence, though it should be right here. Okay. And then in the sequence number 7, there's no gap. And then in the sequence 8, we have a gap in the wrong position. And in the sequence 8, 9, we have also 2 gaps at the end. So here, wrong position. That is the issue with this sequence alignment. And that comes here in particular, the failure of the alignment comes from the fact that the percent of the mutation is very high for this sequence. So normally we have like mutations coming in the order 1 per 100 or something nucleotides, and here is the 1 per like 3-4 of them. So therefore there's less information in the alignment which can be used by the algorithm. So, but that is the general problem, and I believe we have to be aware of this problem. My sequences, including parental one, so in their original form, so fully aligned as they come. So now gaps have phylogenetic meaning in these positions, though they still result into the obliteration or some reduction of the phylogenetic signal. You will see the consequences of this. Okay, so I will start with my phylogenetic analysis using this data. And then first have a look, of course, at the p distance. Let's see at the p distance. So I choose p distance, genetic distance between them, and I calculate these pairwise distances. So this is my matrix. And the parental sequence number 1 should be equally distanced from all of these sequences, because there are 3 generations. So, and the number should be like about 0.3%, 30%, 33% for all of the generated sequences. And you can see they're not. So, and that happens because of the erosion of the phylogenetic signals or presence of the gaps. See how devastating the gaps effect of the missing information might be on the genetic distances. So parental should be 3 generations away from this. Sequence 1 should be 3 generations away from parental in here. So this should be 033. No. So what about this? No, 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 no. So all of these sequences are now biased. And let's see the effect of these gaps on the phylogenies. So let's start with the maximum likelihood method. Here, don't even need bootstrap. So, and I use all sites, including gaps. So here is the result. So we identify our parental sequence here. So then I put the root to parental sequence, because maximum likelihood trees are not rooted. So right here. Okay, so now my parental sequence is an outgroup for all of these sequences. And now we can see 1, 2, 3, 4. Okay, this comes relatively okay-ish together. So 3 and 4 together. So then in this group, the 8 sequences grouped together with the 1, 2, and 3 instead of with the 7, 5, 6, and 7. Also 7 and 8 should be together, but they are not. And then 5 and 6 should be together, and then they are not. You see the devastating effects of the missing information inside. Okay, so at that point, I would like to ask you to analyze your data. So your own data, the ones which you generated during playing the game. Please analyze the genetic distance matrix, see whether it's consistent, how consistent is with your expectations of the matrix. So please construct expected matrix and then see how your matrix differs or deviates from the expected. And also with these sequences. So how the sequence, the phylogenetic trees coming out in your particular reconstructions in comparison to what you would expect from it. Don't forget the parental should be included and the trees should be rooted. In the next session, in the last section of this lecture, so I will show you how this machinery of phylogenetic analysis can be applied to the problem of the origin of coronavirus.